happy Monday. But kahit na happy ating Monday, we have a lot to discuss because I said chapter 12 is quite lengthy. So for today, actually, we'll be continuing our discussion na, no, on the roles of your endoplasmic reticulum, no, and your Golgi apparatus when it comes to protein trafficking from your ER to your Golgi, punta to outside, right? And this will also usually involve a process which we call exocytosis, which we will be talking also about in a while. Okay, I do have some reminders towards the end of the class, so please stay put. But for now, let's discuss and continue discussing rather, yung ating trafficking throughout the membrane system. Okay, to have a refresher of what we have discussed in the previous lecture last week, remember that yung ating mga proteins that were synthesized, right, in your rough endoplasmic reticulum, di ba? Take note of the difference between the two, rough and smooth. Di ba? Smooth doesn't synthesize protein, only the rough endoplasmic reticulum does. Di ba? So those proteins that are synthesized in your rough endoplasmic reticulum, they are going to be eventually sent, diba, to your different target locations, different intracellular locations that will include, for example, the ER itself, the Golgi apparatus, and also yung mga affiliated natin ng mga organelles like the endosomes and the lysosomes, diba? And once it reaches, yung target organelle niya, where it is eventually to remain there kasi yun na nga yung kanyang target location eventually, there needs to be a mechanism, that will prevent the protein from leaving. So once na ma-reach yung area na yun, dapat doon lang siya. It cannot go back. So hence, what the system does, is that each of these proteins will have what we call these specific tags, and these specific tags are very important, for the protein, na ano siya para siyang quote and quote marker, for the protein that is with it, as it goes, from one location to another, so by targeting it, it will be part of the transport vesicle that will take it to the correct location. So pag markado siya, na okay, this because of this specific tag, alam ng system na it should be sent to your Golgi apparatus. It knows that it should be sent out for your endosomes. It should be sent out towards another organelle, diba? And we will be discussing what types of tags are, what those tags are, So depende yan in its, let's say, the protein, depending on the location that it will be sent to, that tag can be of different forms. It can be an amino acid sequence, it can be an oligosaccharide side chain, hydrophobic domain, etc. In addition, yung mga membrane lipids, they also be tagged, to help vesicles reach their destinations. So lahat sila dapat merong associated, na marker tagging for the proper sorting and direction later on. Okay? So like we said, these tagged proteins, these tagged polypeptides that are encoded by the nuclear genes are going to be routed to your different locations, different compartments of your eukaryotic cells. For example, in the endomembrane system, cytosol, mitochondria, chloroplast, peroxisomes, interior of the nucleus, etc. Okay? But in all of these cases, yung process will obviously begin with the transcription of, again, DNA, diba? DNA into RNA that are processed in the nucleus, and then they will be moved through nuclear pores for translation in the cytoplasm, where most of your ribosomes are going to be found. And once they arrive in the cytoplasm, these mRNAs become associated with your free ribosomes. And shortly after the translation process begins, there are two main pathways, for routing the newly formed polypeptide products. No? And these two are we will be discussing in depth. No? The first pathway is what we call the co-translational import, okay? And this is utilized by ribosomes synthesizing polypeptides that are destined for export, no, from the cell. As you will see later kasi yung other mechanism, it has a different target location, no, and target function. For this one, this is used for those that are going to be exported, no, palabas ng cell. Okay? So in this kind of process, in your post-translational import, the movement of your polypeptide across or into the endoplasmic reticulum membrane is directly coupled to the translation process. Kaya nga siya tinawag na co-translational. It is coupled directly to your translational process. No? Meaning, polypeptides, no, or transferred across the ER membrane as the synthesis is happening. No? Sabay silang nag-o-occur. And the other pathway that I have mentioned earlier is the post-translational import. Okay? And this is used for polypeptides that are destined for specific organelles like the cytosol or the mitochondria, your chloroplasts, peroxisomes, or the interior of your nucleus. Okay? So for this one, yung mga polypeptides that have been finished with the translation process, they are released from your ribosomes and they remain inside the cytosol, or they can be taken up by an appropriate organelle later on, like any of these ones, no, with the presence of your special targeting signals. We'll be getting back to them in a while now, those both mechanisms. But this is a general overview of what you can expect, again, from your membrane trafficking throughout the endomembrane system. Remember last time, di ba? We have mentioned yung process from your ER to your CGN phase, to your TGN phase, and towards outside of the membrane. Lagyan natin ngayon ng mas additional, no, na mga details. Okay? So the first thing, no, is you have yung ating mga ribosomes of your rough endoplasmic reticulum here, di ba? Ito yung ating rough endoplasmic reticulum that has studded, no, ng mga ribosomes, no? So these basically synthesize yung mga proteins, and these proteins will enter the ER lumen where magkakaroon tayo ng initial steps for the glycosylation that will happen. Okay? And then you have here mga transition vesicles, right? And these will carry yung glycosylated proteins and newly synthesized lipids. Saan siya dadating? Dito, di ba? Sa ating CGN phase, sa cis phase. Because remember, the cis phase is the nearest one na phase of the Golgi facing towards your rough endoplasmic reticulum, di ba? So it will now arrive to your CGN phase. Okay? And then these lipids and proteins, di ba, meron tayong two modes of movement, di ba? Anterograde at retrograde. For CGN to TGN, it will move in an anterograde process, di ba? Because it carries anything that is from your transition vesicles arriving in the CGN, it goes to TGN through the anterograde process, no, or transport. But we mentioned also, di ba, meron tayong two models of how this can be transported. Meron tayong stationary cisternine model, di ba? At saka yung maturation model, di ba? Either way, yung pag-carry niya will be through anterograde transport, di ba? As it moves either through the cisternine, di ba? Or nagmamature yung cisternine natin, or nag-fuse and nagbabud off from one cisternine to another. Di ba? Remember that. And then, once it arrives at the TGN, it will form some of your secretory vesicles, di ba? And as the name implies, kaya nga secretory ang name niya, it will release the contents at the plasma membrane by exocytosis. Okay? So, once na na-release na niya, so it's bound, no, for any of your target organelles or target locations. Okay? But, there will be instances, di ba, that yung nasabi nga natin, not everything will be totally used. Di ba? Some of it, for example, will be involved in the formation, no, o itong mga secretory vesicles, no, vesicle buds. You can see here itong mga secretory buds, yung iba secretory vesicles, ito ibang mga buds na to, vesicle buds, they will form, no, yung mga endosomes natin, no, to help make yung itong lysosome. So, at the same time, no, yun nga mga proteins and other materials that can be taken into the cell by endocytosis naman, no, which we'll be discussing in a while. Kasi di ba kanina exocytosis, exporting outside of the cell, di ba? Eh meron tayong mga certain proteins and materials that can be taken into the through endocytosis, no? So, dyan ngayon papasok yung role, no, ng mga vesicular buds natin, which helps the formation of your endosomes. Because these vesicles, no, will fuse with your early endosomes, no, that will carry yung mga entering na materials, no. Pwede siyang makarry papasok, no, for let's say digestion, ganyan, for formation later on, no, ng mga late endosomes and eventually ng mga lysosomes, diba? And lastly, no, itong mga lysosomes natin, no, will become part of again the system, no, and these some of the materials here during the late endosome will again fuse with your cisternae to return, no, to your earlier compartments and become part of it again in order to recycle some of its parts in a retrograde manner. Okay? So you will see, no, that there are specific mechanisms for our vesicles, no. Once it is exported, no, meron tayong specific mechanism for that. Meron tayong mechanism for those entering, no, or those that are being recycled, no. It will involve endocytosis and exocytosis. Okay? I hope this mechanism is clear for everyone. Okay? So that's the general overview, no, of our mechanism, no, of your trafficking. So let's now discuss each detail, no, of how again our pathways that occur for protein products diverge. Okay. First one is with your co-translational import. Okay. So for this mechanism, co-translational import into your endoplasmic reticulum, this is the first step, no, in the pathway for, again, delivering newly synthesized proteins to your different locations within the endomembrane system. Okay? So, specific siya, no, for within the endomembrane system. Okay? Kasi di ba, yung ating post-translational import, yun yung pupunta to other targets, di ba? Like yung mga mitochondria, chloroplast, etc. Okay? So within the endomembrane system, no, for itong post-translational import, the proteins that are synthesized in this way are handled, no, where yung ribosomes are being attached to the ER, no, shortly after the beginning of translation. So the proteins are synthesized on the ribosomes that become attached to the ER shortly after the beginning of translation. And the polypeptides synthesized on your rough endoplasmic reticulum are localized inside the ER lumen. So makikita nyo, no, that the ER, no, once they enter the ER, they are also being synthesized at the same time. Kaya nga ang pangalan niya is co-translational. It is being synthesized as is it being entered, no, as it enters rather, no. And they were able to show this mechanism, no, with a radioactive labeling experiment that showed yung mga newly formed polypeptide chains being inserted through your endoplasmic reticulum membrane as they are being synthesized na. No? And there is an antibiotic that they use, no, yung puromycin, that causes the polypeptide chains to be prematurely released into the vesicular lumen. So if, let's say, no, yung mga polypeptides move, di ba, into the lumen of your endoplasmic reticulum as they are being synthesized, how does it now determine, no, the cell? How does it determine which polypeptides are targeted in this way? So they were able to figure it using this experiment carried out by Blobel and Sabatini, no, who first suggested what we call the signal hypothesis. Okay, so Blobel and Sabatini, in their hypothesis, signal hypothesis, they proposed that for polypeptides that are destined for the endoplasmic reticulum, yung N terminus, di ba, of that, no, will contain an ER signal sequence. And this ER signal sequence is responsible for directing the ribosome mRNA polypeptide complex to the surface of your rough endoplasmic reticulum, where the complex docks, no, or anchors at a protein docking area on the ER surface. So basically, they're telling, no, na merong parang sequence, no, signal embedded within the polypeptide, no, that triggers, no, that tells what to do. Okay? They also further mentioned, no, that as the polypeptide chain, di ba, nag-elongate siya during the mRNA process, it continues or progressively crosses yung ER membrane and enters the ER lumen. No? And upon passage of the newly synthesized na mga protein across the ER membrane, nag-dissociate yung ribosomes from the site of the synthesis, and they re-enter the cytoplasmic pool, which now signals the start of another round of synthesis. We'll discuss this more in the next slides. Okay? So shortly after proposing that, Milstein and his colleagues, no, found an evidence that there is a 20 amino acid sequence segment that functions as an ER sequence, no? ER signal sequence. And itong signal sequence na to is removed, no, from the polypeptide when it moves to the ER surface. So nawawala din to, nagdi-dissociate din tong signal peptide na to once it moves into the endoplasmic reticulum, no? And subsequent studies revealed that other polypeptides destined for your endoplasmic reticulum will also possess the N-terminal sequence that is required for targeting the protein to your endoplasmic reticulum, and that is removed, no, as the polypeptide moves into your ER. So proteins containing this kind of ER signal sequence at their N-terminus are what we call the pre-proteins. Okay? And yung mga polypeptides with the ER signal sequence, sila lang yung pwedeng ma-insert, no, into or across the ER membrane as their synthesis occur. So you can see the importance of having this signal sequence, no, as their synthesis and their entry to the ER occurs, no. Contrary to the original signal hypothesis na na-mention natin earlier, the ER signal sequence is not the one that directly initiates, no, the contact with your endoplasmic reticulum. Instead, that contact, no, with your polypeptide in the ER is mediated by what we call the signal recognition particle, or SRP. No? And this one, the SRP, recognizes and binds to the endoplasmic reticulum signal sequence of your newly forming polypeptide and then binds to the ER membrane. No? So you will see that the signal recognition particle is separate, no? Is a separate entity from the signal peptide that we have mentioned earlier, but rather it makes a complex with it, no, later on, and binds together with your ER membrane, no, to allow for the process to occur. Okay, so let's illustrate, no, given yung ating SRP, given yung ating signal sequence, etc., and yung ribosomes, let's discuss, no, how the SRP, no, plays a role in your co-translational endpoint. Okay, so let's discuss each of these figures one by one. Okay, so our process here, it begins when yung ating mRNA encoding polypeptide destined for your ER starts to be translated, diba, on your free ribosome. This big green thing, no, turquoise green thing, is yung ating ribosome. Okay, so think of it as the ribosome. So polypeptide synthesis divide proceeds until the endoplasmic reticulum signal sequence has been formed. And then, you have here, ano ulit itong SRP? Ito yung ating signal recognition particle, di ba? So this SRP, as we have mentioned in the previous slide, it binds to your ER signal sequence here. Okay? Ito yun. Ito yung ating ER signal sequence, no? And when it binds, it blocks yung further translation of your polypeptide. Okay? So the SRP binds now with the ER signal sequence, no? And the SRP also binds the ribosome to a structure in the ER membrane, which we call the translocon. Okay? So this translocon, okay, you will see, di ba ito na yung SRP natin, di ba? The SRP that is bound to your ER signal sequence, di ba? So in a sense, parang isa na siyang complex here. Okay? And itong buong complex natin of your signal sequence and the SRP receptor, it bounds again to your translocon and it makes another complex. Okay? So this translocon now is composed of your again yung ating ribosome receptor, no, that holds the ribosome in place. You will see here, no. It also has the pore protein that you can see here, yung pore protein that forms a channel, no, yung opening through which the growing polypeptide can enter the lumen. Kasi dito, as you will see here, no, ito yung pore protein kung saan yung ating polypeptide can enter later, no. And you also have the signal peptidase, which is an enzyme that removes the ER sequence. So ngayon, it's a complex of four things, di ba? Yung ating SRP complex to the signal, you have the ribosome receptor, you have the pore, and you have the signal peptidase attached to your ribosome receptor, no? That will remove the ER signal sequence. Okay? So now, no, the SRP, you will see with each attached ribosome, naka-attach siya dito. Okay? It binds the SRP receptor that allows the ribosome to bind the ribosome receptor. So nag-bind na silang lahat ngayon. Diba? And this will now allow also the GTP, okay, that it will allow to bind, no, both yung ating SRP and the SRP receptor, as you can see here. And once na mag-bind ang GTP, it will allow for the unblocking. Di ba kanina kasi nag-block eh, nung nag-bind yung SRP doon signal sequence when it attaches also to the ribosome. So now, kapag may GTP ngayon, it unblocks the translation process and you will see here, cause the transfer of your signal sequence to your pore protein. Remember the pore protein, ano yung ginawa niya? Yung channel, di ba? So now, makakapasok, di ba? Yung ating signal sequence through here, no? And the polypeptide can be inserted now. Okay? The signal sequence together with the polypeptide can now be inserted. So later on, yung GTP natin, ma-hydrolyze siya. And once it hydrolyzes, magre-release din yung SRP natin. Okay? And as this occurs, yung polypeptide natin will continue to elongate. No? You will see here, it continues to elongate and it passes into your endoplasmic reticular lumen where yung ating signal peptide cleaves the signal peptide. It cuts sa signal peptide itself. No? So this signal peptide, once it is cleaved, di ba, inact on na kasi na signal peptidase, magde-degrade na siya, no? In the ER lumen, mawawala na siya. Okay? So once the polypeptide synthesis is complete, this polypeptide will be released into the lumen, no? And the ribosome will detach from the ER membrane. And itong subunits, no, will also dissociate and release the mRNA. So that's the process, no, of your co-translational import. Okay? Most of the proteins that are synthesized, no, on your ribosomes attached to your ERs, no, are glycoproteins, which we mentioned last time, di ba? And ano uli yung glycoproteins? These are proteins with covalently bound carbohydrate groups. Okay? So for the initial glycosylation na mga reactions, it occurs in the ER often while yung growing polypeptide is being synthesized. Kaya nga co-translational, di ba? So after polypeptides have been released into your endoplasmic reticular lumen, yung after it's also been glycosylated and also after being folded, they are delivered to your various types of transport vesicles to their destinations. And their first stop is the Golgi apparatus, where further glycosylation and processing of your carbohydrate side chains may occur. Okay? So the Golgi apparatus will then serve as a site for sorting and distributing proteins to your other locations. For proteins that are, let's say, soluble in nature, yung default pathway niya, it is from the Golgi apparatus, no, to your secretory vesicles that move to the cell surface and fuse with the plasma membrane, or leading to your secretion. Okay? Those that are not destined naman for secretion have specific side chains, no, or signal sequences that tag them, that target them to destinations within the endomembrane system. So iba, di ba? Yung mechanism for soluble proteins for secretion as compared for non-secretion, di ba? So like we mentioned, different signaling mechanisms will be used, no, for proteins for those who have a different destination. No? Proteins din na ang final destination is ER, iba din yung mechanism. No? So for this one, proteins that yung kanilang final destination is the ER, they have what we call the K-del sequence, which is basically a representation, no, of your amino acids lysine, as, glu, leucine, diba? Or any related sequence. Okay? So the Golgi complex now will also employ a receptor protein that binds to your K-del sequence and will deliver the target protein pabalik ng ER. So you will see, having, for example, soluble protein that is for secretion, we will be discussing later on ano yung tag niyan. Yung mga secretions, non-secretion proteins naman, no, within the endomembrane system, iba din ang kanyang tag niyan. And yung babalik ng ER, K-del ang ginagamit naman nila na tag, di ba? So ito yung sinasabi natin na other tags, no? So for example, yung mga proteins, no? Protein composition in the ER, let's say. Pag kunwari, kailangan nilang uh, ma-maintain or they need to be retained, maintained within the endoplasmic reticulum, iba din yon. Iba rin yung protein, na protein tag na kailangan for those destined for plasma membrane. Iba rin yung for Golgi to ER, no? So for example, some protein tags, no, or proteins that are localized to the ER, no, contain the sequence RXR, no? This is what we call the retention tag, no? RXR being R, your arginine, and arginine, dalawa, no? And X is basically any amino acid, no? So this being a retention tag, this is found in some proteins that are also destined for plasma membrane, no? And this RXR tag must be hidden or masked, no, to allow the assembled protein or complex to leave the endoplasmic reticulum. Okay. So dalawa siya. It's either for being localized to the ER or it could also be those for our destined sa plasma membrane. Okay. While yung ating mga retrieval tags, no, are these are being used for proteins that are returning, no, from the Golgi to the ER. So let's say yung protein, when this tag binds to a receptor, yung receptor ligand complex is packaged into a transport vesicle for return pabalik doon sa ER. And these tags are usually short C-terminal amino acids like yun nga, yung mga kidal natin kanina or yung KKXX, no? Lysine or XX being any kind of amino acid, no? And this can be found in mammals, kung kidal or KKXX, while yung HDL, his as blue leucine, no? This is found in yeast, okay? And these are used again for those returning from the Golgi to your endoplasmic reticulum. And then there are also some proteins, no, of the Golgi apparatus. Kasi kailangan minsan yung proteins na yon, they don't leave the Golgi apparatus. Like similar doon sa endoplasmic reticulum, there are proteins that are only required na resident siya doon, resident protein siya ER. Ito rin, may mga Golgi complex proteins din tayo na for Golgi lang dapat talaga siya, no? So they will also contain similar tags, no? Or retention tags or retrieval tags, no? So, in addition to that, yung mga Golgi apparatus, like those in the ER, the formation, no, of large complexes that are excluded from the transport vesicles may play a role in maintaining the protein composition of the Golgi complex. Okay? And in addition, a distinctly different mechanism is also likely to work for your Golgi, no, which is a mechanism involving hydrophobic regions of Golgi proteins. So your Golgi proteins, no, all Golgi specific proteins are considered to be integral membrane proteins, no, with one or more hydrophobic membrane spanning domains. And their length of the hydrophobic domain may determine into which cisternae protein is incorporated as it moves through the organelle. And among your Golgi specific proteins, there is a corresponding increase in the length of hydrophobic membrane spanning domains going from your CGN phase to your TGN phase. And these proteins tend to move from compartment to compartment until yung thickness of the membrane exceeds the length of their membrane spanning domain, hence blocking further migration. And during their journey through the ER and early compartments of your Golgi apparatus, you have soluble lysosomal enzymes like yung mga glycoproteins that undergo glycosylation and glycosylation, for example, followed by the removal of glucose and mannose units. So within the Golgi apparatus, yung mannose residues also on the carbohydrate side chain of your lysosomal enzymes, they are phosphorylated and forms an oligosaccharide containing a mannose 6 phosphate. So this oligosaccharide tag now distinguishes soluble lysosomal proteins from other glycoproteins and ensures their delivery to your lysosomes. So let's use all of those um, ideas, no, in explaining uh, the targeting, no, of your soluble lysosomal enzymes to endosomes and lysosomes by your mannose 6 phosphate tag. Okay, so going from top to bottom, no, in your endoplasmic reticulum, you have yung mga soluble lysosomal enzymes, no, that undergo and glycosylation, followed by the removal of your glucose and mannose units. Okay. So the hydrolytic enzymes destined for the lysosome are phosphorylated on a mannose residue, and the tagged lysosomal proteins now bind to your mannose 6 phosphate receptor. Yung receptor natin na yun, no? And then, yung ating receptor ligand complex, they are now being packed into a transport vesicle and conveyed rather, no, to an endosome. Okay. In your animal cells, lysosomal enzymes are transported from the TGN to your organelles known as your late endosomes. Diba? And the dissociation of these lysosomal enzymes prevents the retrograde movement of the enzymes back to the Golgi, no, with your receptors. So these late endosomes, they mature to form a new lysosome, or it can deliver its contents to an active lysosome. So babalik. So far, no, we discussed the co-translational import and sorting of your soluble proteins that are destined either for, let's say, secretion palabas ng cell, no, or for the lumen of the endomembrane components. The other major group of polypeptides that are synthesized on your ER-attached ribosomes consists of molecules that are destined to become integral membrane proteins. Remember, no? Ano ba ibig sabihin natin ng integral membrane proteins? They are integral because they are anchored, di ba, to your lipid bilayer by one or more alpha helical transmembrane segments. Okay? And there are two main mechanisms by which hydrophobic transmembrane segments anchor these newly formed polypeptide chains to the lipid bilayer of your ER membrane. So paano ba naa-anchor itong mga integral membrane proteins? The first mechanism will involve polypeptides with your ER signal sequence at the end terminus, which allows the SRP to bind the ribosome mRNA complex to the ER membrane. So what happens here is that yung elongation of the polypeptides, as you will see here, no, it continues, no, to elongate, humahaba siya ng humahaba, no, until the hydrophobic transmembrane domain is synthesized. So the hydrophobic amino acid functions now as a stop transfer sequence, no? And this basically stops, no, halts the translocation, yung pag-move from the cytosol to the lumen, no? It stops the translocation through the ER membrane. So as your translocation then continues, yung rest of the polypeptide now will stay no on the ER membrane. So basically no, kunyari, 'di ba? Ito yung ating ER signal sequence, 'di ba? So mag-elongate yan, mag-elongate, 'di ba? But once na yung stop transfer sequence natin, no, is being encountered, magbabind yan, no, to our channel, 'di ba? So magsa-stop na rin yung continuous niya na pagpasok sa ER lumen, no? Pero yung elongation niya magko-continue pa rin, pero dito na lang sa labas. Kaya yung cytosolic end of the polypeptide will be, let's say, longer in this case, no, as compared to those in the ER lumen, no. And then once na yun nga mag-continue yung production of the polypeptide, eventually yung stop transfer sequence natin, it moves laterally, no, forming a permanent transmembrane segment. So now, anchored na siya. Kita niyo, di ba? To your bilayer. One of the bilayers. The second mechanism will involve membrane proteins naman without a typical signal sequence at the end terminus. And instead, these proteins have an internal starch transfer sequence. No? So this internal starch transfer sequence is where the SRP binds to and targets to the ER membrane. So this hydrophobic region na after it binds to, yung hydrophobic region niya functions now as a membrane anchor, no, that binds or once after it binds, diba, yung membrane anchor moves out through a side opening, lumalabas siya, moves to a side opening in the translocon, and it permanently embeds itself, yung polypeptide, in the lipid bilayer. But what if those integral membrane proteins naman na transmembrane na multipass, no? So meron din naman silang ibang mechanism. So for transmembrane proteins with multipass transmembrane spanning regions, they are formed in a similar manner to that of the single pass transmembrane protein, where it uses the start and stop transfer strategy. Except for this, nagkakaroon tayo ng alternating pattern of start transfer and stop transfer. So kapag meron tayo ng alternating na start and stop, it creates a polypeptide containing now multiple transmembrane segments that pass back and forth across the membrane. So magte-thread yan, magte-thread yan continuously, ganyan for example, until ma-encounter niya yung next na stop transfer sequence, di ba? So at this point, yung next transmembrane section is now released into your ER membrane. Once a newly formed polypeptide has been incorporated in your endoplasmic reticular membrane by one of its preceding mechanisms discussed, it can either remain in place to function as an ER membrane protein, or it can be transported to other components of the endomembrane system. So this now will be carried out, yung transport na yon, by a series of membranes that buds and fuses those events into your membrane vesicles as they pinch off from one compartment of the endomembrane system and fuse with the other compartment. So those that we have discussed earlier pertain to their co-translational import. But as we have discussed, paano naman yung mga proteins that are made for mitochondria, chloroplast, peroxisomes, interior of your nucleus? This is now being managed, no, by your post-translational import mechanism. Eh, so, because tong mga proteins na to are synthesized on the free ribosomes and released into the cytosol, each protein must carry a target signal now, no, that directs it to the correct organelle. So, meron silang tag that says this is for mitochondria, this is for chloroplast, etc. No? So, a recently synthesized or translated protein, a polypeptide rather, no, initially associates with what we call the HSP70 molecule, no? HSP70 chaperones. No? And this allows for the polypeptide to keep its unfolded form. Okay. So the polypeptide, no, will then dock with your translocator here, whose core is what we call the SEC61 complex. No? Ito yung ating translocator. And then you have the HHP70 proteins. They will also now be released, no, as it begins to thread through the pore. So as it goes inside and outside, later on makikita niyo yan. And then we have here a complex, no, of your ER membrane proteins, the SEC62, 63, 71, and 72 complex. So this complex is associated with your SEC61 core, and it recruits what we call the BIP, no? Yung ating resident chaperone sa ER, no? It recruits the BIP to the end of the polypeptide, okay? And the BIP pulls the polypeptide into the lumen. So once the BIP molecules binds and release the polypeptide as it enters the matrix here, ang hinihilin niya papunta sa ER lumen using a ratchet-like mechanism. Ultimately, materials in your vesicles are destined, no, for either, again, secretion, eventually leaving the cell. No? And there are two methods that are unique to your eukaryotes for transporting materials across the plasma membrane. These are exocytosis and endocytosis. Exocytosis is the mechanism by which your secretory vesicles release the contents outside of the cell. While endocytosis internalize your external materials. For the next few slides, no, we'll be focusing on the exocytosis because for the next meeting natin ifo-focus naman ang endocytosis. Okay? So one key feature of the vesicular traffic, as we have shown, no, during the overview of trafficking, or the presence of your secretory pathways. No? And these secretory pathways is the mechanism by which the proteins move from your ER through the Golgi apparatus to your secretory vesicles and secretory granules, which then discharge their contents to the exterior of the cell. There have been experiments, no, that have shown how the movement of the proteins occur through secretory pathways, and they did it by radioactively labeling, no, yung mga proteins na yan very briefly, and you will see in a time-dependent manner, no, yung movement of those proteins from one location to another. So for example, yung mga radioactively labeled na proteins, at around 3 minutes, yung mga proteins, they can be seen here localized in the endoplasmic reticulum where it is being synthesized, not just synthesized. A few minutes later, around the 7 minutes, the proteins began to appear. Natatrack nila yun eh. They were able to appear now in the Golgi. ito, here in the Golgi complexes. Then after 30 minutes, they saw that the proteins was in the vesicles already, no? That are budding from the Golgi. And at around 170 minutes, the proteins begin to accumulate in your dense zymogen granules and the vesicles, which are basically the vesicles discharge, no? The contents out of the cell. And over the course, no, of many years, we also were now able to distinguish, no, several modes of secretion by your eukaryotic cells. So we have three main modes, no, of exocytosis. You have the constitutive secretion, which involves the continuous discharge of vesicles at the plasma surface. We have also the regulated secretion that involves the controlled rapid release that happens in response to an extracellular signal. And we have the polarized secretion that involves the secretion at one specific end of your cell. And these kinds of secretions can be found in different kinds, no, of cells. Constitutive secretion, more related to, let's say, mucus secretion by intestinal lining. Regulated secretion will involve yung mga neurotransmitters, insulins, and zymogen release. While polarized secretion will involve intestinal digestive enzymes and nerve functions. So let's discuss each, no, of these secretions, starting with constitutive secretion. Okay. So after, let's say, nag-bud off na, di ba? From the TGN phase, yung ating vesicles. These vesicles will now move directly to the cell surface and immediately fuse with the plasma membrane. Okay. So this whole process is unregulated, no? It is continuous, is independent rather, it is independent, no, of external signals. Kasi independent one is regulated secretion. So constitutive is independent of any type of signal. No? So like we've mentioned, this example can be observed in the mucus secretion of your intestinal lining. Previously, biologists thought, no, that the constitutive secretion was the default pathway, no, for protein synthesized by your rough endoplasmic reticulum. No? It was previously thought that proteins destined to stay in the endomembrane system must have a tag, no, to avoid constitutive secretion. But current evidence suggests that some tags may be required for constitutive secretion. In comparison to your vesicles, no, that undergo constitutive secretion, meron tayong mga secretory vesicles naman involved in the regulated secretion that accumulate in the cell and only fuse with the plasma membrane in response to specific signals. So this is what occurs during regulated secretion. An important example of this one is your neurotransmitter release and the release of insulin from pancreatic beta cells in response to glucose and the release of your zymogens, no, from pancreatic acinar cells in response to calcium or endocrine hormones. So regulated secretory vesicles form, again, no, by budding from the trans-Golgi network phase, no, and they are formed as initially immature secretory vesicles, which undergo a subsequent maturation process. Okay? So the maturation of your secretory proteins involves their concentration, which is called the condensation, and sometimes proteolytic processing. So once na nag-mature na yung mga secretory vesicles, They move close to the site of secretion and they will remain there until they receive a signal that triggers the vesicles to release their contents by fusing with the plasma membrane. And lastly, we have the polarized secretion. No? And in many cases, no, yung exocytosis of specific proteins is limited, no, to a specific surface of the cell. This is what polarized secretion is. And this is observed in yun nga, intestinal cells that secrete digestive enzymes only on the side of the cell that faces into the intestine. This is also observed in nerve cells which secrete neurotransmitter molecules sorry, only at junctions with other nerve cells. Ultimately, the material in your vesicles destined for secretion must leave the cell. No? And this is what happens during exocytosis, no? Which are proteins or other materials in a vesicle are released, no, to the exterior, no, of the cell as the vesicle fuses with the plasma membrane. So animal cells, they secrete, di ba, mga hormone, mucus, milk proteins, digestive enzymes in this way. Plants and fungal cells naman, they secrete enzymes and structural proteins, no, for the cell wall. You can see here, no, may mga structures tayo which we call the SV, the secretory vesicles that approach the plasma membrane here, no? And you can see this is where one vesicle has fused already. Ito, no? One of your secretory vesicles already fused with the membrane and its contents have already been secreted out. Sorry, like this one. No? So when this kind of vesicle fuses with your plasma membrane, what happens is that, sorry, yung inner membrane, your luminal membrane of your vesicle becomes part of the outer surface of the plasma membrane. Sorry. So yung glycolipids and glycoproteins that were formed in the ER and Golgi lumen will face the extracellular space. Sorry, class. Okay. So how does exocytosis occur? So you have here, you can see yung vesicles natin, di ba? That contain itong mga products for secretion. Okay? So they move towards the cell surface, okay? And the membrane of your vesicle, fuses. You can see here, it fuses with your plasma membrane. So this fusion of the plasma membrane leads to the discharging of the contents of the vesicle later on. And this membrane, no, of the vesicle eventually becomes part of the cell membrane. Fusion of your regulated secretory vesicles with the plasma membrane is generally triggered by an extracellular signal. And in most cases, hormone or neurotransmitter binds receptors on the cell surface and triggers a second messenger inside the cell. Sorry. And then you have an increased, no, transient elevation in calcium, which we thought to be an important step, no, in signaling cascade that leads to the activation, no, of protein kinases whose targets protein are component of either the vesicle membrane or the plasma membrane. So we will be encountering this more in our lesson towards signal transduction, no? Pero you will see that calcium serves as a trigger for many signal downstream mechanisms, no? Which also involves like this one, yung exocytosis. just an overview no, of what endocytosis, because we will be discussing this in the next meeting, as compared to your exocytosis, diba? Endocytosis will involve carrying the contents from the exterior of the cell towards the interior of the cell. So during endocytosis, there is a small segment of the plasma membrane that folds inward. Then, once na mag-enter na yung mga contents that will be transported to the inner part, nagpipinch off siya. It eventually closes. No? It eventually closes. And this vesicle no, pinches off to form an endolytic vesicle. Okay? It contains the ingested substance or particles. So obviously, no, in terms of membrane flow, exocytosis and endocytosis will have opposite effects. Exocytosis adds lipids and proteins to the plasma membrane, endocytosis removes them. So you will see that there is a steady state composition of plasma membrane resulting from a balance between exocytosis and endocytosis. Na baka kasi ma-overwhelm na kayo kapag tinuloy ko pa ang endocytosis. So, gusto niyo ba ituloy ko pa ang endocytosis? Silence means yes. So mukhang gusto niyo pang ituloy. Kasi we still have enough time to continue that. Sige, wag na. Baka ma-overload na kayo with the information. So, we'll continue our discussion on endocytosis next meeting. Okay? But I do have a few reminders before I let you off and before I take your attendance. As I have mentioned, no, in our upcoming meeting this Thursday, after discussing chapter 12, the remaining of chapter 12, medyo mahaba din kasi pa ang chapter 12, no? So, I'll divide na lang, no? Chapter 12 into two discussions, okay? Chapter 12, after the first discussion there ng ating meeting on Thursday, we'll have a quiz. If you saw our announcement on the course Animal Space, ang quiz natin for Thursday, this Thursday, is on chapter 7 and chapter 8. Okay? So please make sure to bring your notebook, your laptop, your tablet, or your phone, no, for the quiz, because this is online naman yung quiz. Through course Animal Space naman kasi siya, no? And then anything that is left to discuss for chapter 12 will be carried over next meeting, no, next meeting rather, next Monday on June 15. And the remaining time also will be devoted to quiz for chapter 12. Okay? So in the next coming days, meron tayong dalawang quiz. Okay? Chapter 7 and 8 for Thursday, and chapter 12 na quiz on next Monday. Okay? And again, a reminder that our long exam for covering 1, 2, 4, 7, 8, and 12 next Thursday, June 15, 16, 18. Ha? And this is offline. Okay? This is face-to-face. Okay? Scantron tayo. Okay? So, expect the unexpected. Just kidding. You can expect almost similar to how the quizzes are, no, yung exam. Syempre, but yung forms of questioning, iba lang. So, wala tayong matching type kasi Scantron siya. Diba? So, expect. Okay? Do you have any concerns or questions regarding our discussion today?